Medurstand, Understanding Medicine.
I'm Professor Aziz-ur-Rehman, and after discussing the role of gut microbiota in health, today we are going to discuss the role of microbiota in diseases. And there is a lot of interest in this new science, and uh, I just typed the word microbiome and human disease and see how many papers I was able to pull. 19,940 papers are published on medical journals, and this is the source, the PubMed. So that shows the level of interest in this new science. There is complex interaction between host and microbiome and then the environment. Our environment and host, they have interaction, both have effect on each other, and then host has interaction with microbiome, and this is very delicate balance. Normally our microbiota and ourselves, we live without harming each other, in fact benefiting each other. This is win-win situation. This relationship is mutually beneficial, and we live a normal healthy life. But sometimes, because of some environmental factor, and there could be one of the several environmental factor which can alter this microbiota, and this is called dysbiosis. Alteration in our normal healthy microbiota is called dysbiosis. So dysbiosis then can have it can cause disease because this is a changed environment. The normal balance is disturbed. Now this biosis would promote disease not only in the gut but also elsewhere. But at this stage, it is hard to tell, is it the disease which has caused this dysbiosis, or is it a dysbiosis which has caused this disease? Because we see in an animal or in some individual a disease and dysbiosis. So which one is the cause and which one is the effect? There was an experiment done, and that is actually the proof of concept. Now, if the microbiota from the diseased animal is transferred to a healthy mouse, that mouse develops disease. Now, if that can be shown, that of course would mean that dysbiosis is the primary thing, and that has caused disease, and this dysbiotic microbiota can cause disease if transferred to another animal. And this has been seen in many diseases, particularly in Clostridium difficile infection and autism spectrum disorder, asthma, obesity, many metabolic diseases. There is strong possibility that dysbiosis is actually at least partly responsible for the pathogenesis of these diseases. There are several areas where there is indication that microbiota may be actually linked with our diseases, and these are infections and visceral diseases, and out of viscera, gut, liver, and brain disorders, allergic disorders and autoimmune diseases, and metabolic syndrome and obesity. This is perhaps the biggest medical challenge we are facing these days. And atherosclerosis, again, perhaps the biggest disease of mankind, which leads to heart attack and strokes and gangrenes. So there is possible link uh, uh, of microbiota with these diseases. I'm going to examine them one by one. First, take the infection. Okay, all right. Okay, so the theory is that somebody had normal microbiota till the time that person developed some infection, intestinal infection. That would be perhaps self-limiting, but that person took a course of antibiotic. Antibiotic was given with the intention of killing uh, pathogens, but it also killed healthy microbiota, and that led to dysbiosis. So this dysbiosis would promote those bacteria which are present in our gut, but they remain dormant. But in this environment, in this dysbiotic environment, they get a chance to grow and cause disease. So in majority, this would also be self-limiting, but in some cases, this would go on to develop a severe type of colitis. We call it Clostridium difficile infection. Now, it is not restricted to the intestine only. There are actually some effects to the body also. This is called multi-organ dysfunctional syndrome. So this is how uh, dysbiosis can lead to Clostridium difficile infection and MODS. Can it be reversed? So if this dysbiosis has caused the infection, then the question arises, can, if we restore the bacteria, can the disease be reversed? Of course, we can. Yes, it can be. In fecal microbiota transplantation is currently a standard therapy for those who have developed Clostridium difficile infection and are refractory to antibiotics. They can be treated with FMT, and experiments have shown the success rate in approximately 95% people. Drug-resistant CDI respond to 95% of FMT. FMT can be done through capsule, through nasogastric tube, or through colonoscopy. But actually, the donor's fecal matter is delivered to recipient's gut. But there is a possible downside also. There is possibility that there could be transmission of other infections or unwanted traits. And there is an experiment, an interesting case report I'll share with you where this unwanted trait was actually transmitted. And so we will have to select the donor like we select donor for blood transfusion and organ donation. So I think here also the science will develop how to select the donor to avoid the downside. But the upside is very, very strong, a 95% response rate. So this is proof of the concept that it was the dysbiosis which caused CDI, and once we corrected dysbiosis, the patient responded and healthy flora grew. Infection with H. pylori. H. pylori is again one of the common pathogens. If somebody is unlucky to acquire H. pylori through water or some food, that would lead to dysbiosis and that will cause acid peptic disease. This is actually very common these days. Most patients with duodenal ulcer and gastric ulcer, they have associated H. pylori infection. This can lead on to develop cancer. So it's not just infection, it could actually lead on to develop cancer. So this is again dysbiosis which led to this serious problem. Some other infections, specific bacterial vaginosis may increase the chances of preterm birth. No, I'm not talking about just vaginal infection. Some specific strains of bacterial species, if they are present in somebody's vagina, it could lead to preterm birth. It can lead to acquisition of sexually transmitted infection. Somehow these bacteria, they predispose to acquire sexually transmitted infection, acquisition of HIV infection, then poor response to tenofovir as Gardnerella gobbles up the medicine. So these bacteria can actually eat up the antibiotic, antiviral, so there could be poor response. So this is how many infections are actually linked with these microbiota. The second one is visceral diseases, gut, liver, and brain. We'll examine very briefly. Of course, time will not allow me to go into details, but very briefly, I'll touch upon these three categories. Uh, Gut microbiota is linked with gastric cancer, is linked with colorectal cancer, is linked with Barrett esophagus and esophageal cancer, with Crohn's disease and ulcerative colitis. So I again say they are associated, as the title of this slide says, association of microbiota and visceral diseases. In case this is the gut. So all these diseases are linked with change in the microbiota. Now, which one is cause, which one is effect, we don't know yet. But there is definite association between all these and gut microbiota. This is the hypothesis. H. pylori is considered to be class 1 carcinogen. Now, this has been classified by WHO. That means there's no doubt that H. pylori can cause cancer. Of course, not in everyone, but it has the potential of causing cancer. Then atrophy, metaplasia, dysplasia, that promotes cancer. And this is actually perhaps due to this is the mechanism. These are bacteria, enterotoxigenic Bacteroides fragilis. They produce fragilicin. They reduce this short chain fatty acid. This somehow is linked with these cancers. This one is association of microbiota and visceral disease, and now we're going to take up this liver microbiota gut liver axis. Now, gut microbiota is linked with spontaneous bacterial peritonitis. Of course, you need to have the background condition like ascites, but presence of specific type of gut microbiota will promote the development of SBP and portosystemic encephalopathy, hepatocellular carcinoma, autoimmune hepatitis, and non-alcoholic fatty liver disease. This is very, very common. So I'm not saying that a gut microbiota is known to cause these diseases, but what I'm saying is that we have found the association between these conditions and gut microbiota, whether change in gut microbiota contribute in the development of these conditions or the relationship is reversed, but there is definitely association. The hypothesis is that in dysbiosis, these Enterobacteriaceae, they are increased and Bifidobacterium are reduced in number. And there is production of ethanol, ammonia, and acetaldehyde by these bacteria, and bacterial translocation. There is a kind of increased permeability of gut, and these bacteria or their components, they get translocated into the blood, and there is a kind of endotoxemia and immune dysfunction. This endotoxemia is not in the sense we normally use. This is like saying some endotoxins of bacteria, they circulate in the body and they produce various functions which may contribute somehow in the development of these diseases, because we have found the association. Now the third category, subcategory, microbiota and visceral disease, and now we will take gut brain axis. Now gut microbiota is linked with our eating preferences, the type of food we eat or the amount of food we eat. It is linked with mood, memory, Alzheimer's disease, major depressive disorder, autism spectrum disorder, and multiple sclerosis. Again, I say there is association. Cause and effect relationship is not yet established. This is the hypothesis. Dysbiosis can lead to alteration in serotonin production. Serotonin, is something which controls our brain function. It's a neurotransmitter, and deficiency of serotonin can cause depression. So serotonin works through the vagus nerve and signals go to the brain through the afferent fibers, and that can affect our thinking process and our memory and emotional state. So when we say my gut feelings, we often use this word, my gut feelings. So whenever we say my gut feeling, actually maybe we are talking science, not just a proverb, where it's maybe actually science. So this is how our gut can affect our brain various functions through the vagus nerve. So this was about gut microbiota and visceral diseases. Now we'll take allergic disorders and autoimmune diseases. We are done with these two. Allergic disorders and autoimmune diseases. Gut microbiota is linked with asthma, with allergies and type 1 diabetes. You know, type 1 diabetes is also an autoimmune disease. So these are associated with gut microbiota. This Clostridium sensu stricto that predicts allergic disorders, and there is increase in the production of interleukin 10, and there is reduction in the interleukin 4 and 5 and 13. Bacterial genes, they lead to production of 1,2,1,3 dihome. Exposure to pets and cockroaches in childhood, they reduce the prevalence of asthma. Lactobacillus johnsoni, it reduce the chances of asthma. Early life antibiotics or hyperhygienic condition is actually directly proportional to type 1 diabetes, autoimmune disease, and allergic disorders. Of course, we are not discouraging hygienic condition, but studies have shown that hyperhygienic condition, they actually increase the chances of type 1 diabetes. For example, in our country, the prevalence of type 1 diabetes is 5 out of 100. If you take 100 cases of diabetes, only 5% has type 1. In Scandinavian countries, it is up to 25, 30. So that means, because that is definitely much more hygienic situation than ours. Now, last one, and the second last is metabolic syndrome and obesity. And here I like to examine a few examples. Obesity. Now, gut microbiota is linked with our weight because it affects our eating behaviors, and it also affects the amount of food we intake, and metabolic syndrome and type 2 diabetes, which is the major consequence of overeating and obesity. So let's see, examine this hypothesis in more detail. Firmicutes are directly proportional to the BMI. That means more firmicutes mean higher BMI. That means obesity. Whereas Christensenella is inversely proportional to BMI. So this looks like a friendly bacterium. Christensenella, it reduces food intake and is associated with lean physique, and predictive value is 90%. So imagine, from these microbiota you can predict the physique of the person. Isn't it great? So predictive value 90%. That means the association is very, very strong. And these gut microbiome and is related with insulin resistance. It has been seen that the same food can produce different glucose responses because of different glycemic indices are seen because of different gut microbiota. Certain microbiomes signatures predict type 2 diabetes also, like here, a presence of some specific type of microbiome can predict the future diabetes. Very, very interesting experiment. Now, this is the obese person, and if the gut microbiota from this obese person is transferred to this animal, which is of normal physique, and it has been seen that this mouse would grow in size because of wrong genes, because of obese genes, this animal grew in size. And this has been shown in experiments. So now we are getting closer to the hypothesis that microbiota is actually cause of obesity, not just the association. Now, Reverse experiment has resulted in weight loss. The microbiota from the thin, lean people, if transferred to obese mouse, that has promoted weight loss. So, if that turns out to be true in humans, we are all obsessed about our weight. All of us want to lose weight. So, the microbiota of thin people may be in high demand and they may become filthy rich because of their microbiota selling in high price. Why some people just can't lose weight despite dieting, and we have seen this so many times that people are struggling, they're restricting their diet, they're doing exercise, but they're not losing weight. Of course, we normally attribute to their genes, but it could be their gut microbiota. Gut microbiota may harvest calories from fiber. We consider fiber as something without calories, but in these patients, this fiber may actually be converted to calories. And at least theoretically, it is possible that these gut microbiota may be able to produce calories from air and water. Of course, we have to we always have water and air. So I think there is a possibility that the wrong gut microbiota can actually produce calories from air and water. So those who are obese, they are not to be accused for being fat. It's not their wrongdoing. They may just have wrong genes and wrong microbiota. And those who are skinny, they may not deserve any special credit because they may be thin and slim because they have right microbiota. Of course, right genes and right microbiota. This is the breaking news. It was in one of the journals. This slimy bacterium may fight obesity. This is Archenmensia muciphilia. This is the bacterium which is associated with insulin resistance, uh, sensitivity. More bacteria means better insulin sensitivity. That means less insulin resistance, and total cholesterol is also low. Gamma GT, which is an indicator of fatty liver, that is also low, and WBC count is also low. So this could actually be used therapeutically because this is associated with such good things. And this was actually led to other papers, weight gain after fecal microbiota transplantation. This is very interesting case report. I'll just share with you insight into the role of gut microbiota in obesity, pathogenesis, mechanism, and therapeutic pers perspective. This is the same. Is the same actually. Okay, unintentional weight gain after FMT, fecal microbiota transplantation. So case report. Very interesting. There was a person who had this clostridial infection with Clostridium difficile, which was resistant to multiple courses of antibiotics. And this lady was, I think, 35 years old, and she weighed 61 kg. She was treated with FMT, and the donor was her own daughter, and that lady, her daughter, was 63 kg at the time of fecal uh, microbiota transplantation. But later on, within weeks, this also increased weight. That means that person has the tendency to gain weight. So after FMT got this ICD got cured, but this woman kept on gaining weight, and that woman gained weight, became 80 kg. Her ICD, the infection with Clostridium difficile, got cured, but in 36 months she gained massive amount of weight. This is because of the obese genes, the obese microbiota genes from the donor. Now this actually is very interesting because this is like proof of concept that microbiota can be responsible for development of various diseases and various traits. Possible mechanism for metabolic effects. This is very complex slide published in the New England Journal of Medicine Journal Watch. Now, I have made it easy for you to understand. Specific microbiota that leads to increased permeability and that cause bacterial translocation. Some bacteria they entered circulation and they caused various metabolic effects through endotoxins, and basically there is crosstalk with hormones. Like these endotoxins, these bacterial products, they have effect like insulin, like insulin, glucagon, like peptide, like uh, other peptides like peptide YY. So they actually cause this insulin resistance and obesity and metabolic syndrome. This is the, of course, mostly theoretical, but this is the postulation that how gut microbiota can lead to insulin resistance, obesity, and metabolic syndrome. Hypothesis metabolic syndrome, Escherichia E. coli, they produce a specific type. A specific type of Escherichia coli produces a protein called CLPB, and CLPB has got effect similar to GLP-1 and peptide YY. Either it self has the similar effect or it stimulate the production of GLP-1 and peptide YY. We know these are anorexogenic hormones. They reduce food intake and they increase calorie burn, and that would lead to reduced food intake and increased BMR and ultimately reduction in insulin resistance, metabolic syndrome, and diabetes. So this is the hypothesis. If we can get hold of this Escherichia coli with which can produce CLPB, and if this can be developed in laboratory, this can be used for the management of metabolic syndrome and obesity. The last is atherosclerosis, which, as I said, is one of the biggest diseases of mankind. Uh, let's examine this one. Now, microbiota, again, Escherichia coli specific strains, if somebody harbor these organisms and the dietary phosphatidylcholine and L-carnitine will be converted into trimethylamine N oxide TMAO, which in the presence of atherogenic diet both together will cause atherosclerosis. But if we substitute that person has this microbiota Escherichia coli, but we substitute with this choline dimethyl butanol, and that would not let this TMAO to develop, and in the absence of TMAO, even atherogenic diet would not cause atherosclerosis. So this has been shown. All we need to do is to find out where this microbiota with this capability lives, so we can use that in the management of atherosclerosis. So this was the last component, gut microbiome potential therapies. There are various potential sites. This is almost proven use of stool transplantation for the treatment of drug-resistant Clostridium difficile. I think this is now beyond any doubt a proven therapy. But choline dimethyl butanol can be used for the prevention of atherosclerosis. Bacteroides fragilis can be used for the treatment of anxiety because they produce serotonin, and serotonin is good for preventing anxiety. And then various commensals are responsible for cancers. Maybe we can use them for the cancer prevention. We can manipulate them. Lactobacillus lactis can be used for the treatment or prevention of inflammatory bowel disease, and CLPB producing Escherichia coli can be manipulated to prevent adiposity, diabetes, and a metabolic syndrome. And then bacteriocins So these are actually antibiotics, some specific substances which have antibiotic-like action. They can be used to treat various infection. Now this science is actually going to translate into various types of designer probiotics and prebiotics. Whether that actually materialize or not, time will tell. But there is different potential. Early life probiotics, type 1 diabetes, and asthma. So these are the possible therapeutic options. So summary, everybody has a unique signature microbiome, and it undoubtedly plays a crucial role in our health and disease. Perhaps our life would not be possible without them. And next time you call somebody a piece of shit, you might actually be giving him compliments, because we know that microbiota is so capable and they are capable of doing so many good things. The future might see new lab tests, new therapies based on microbiome, and we might have to rewrite the pathogenesis of many diseases. And in this new science, how much is the hope and how much is the hype is yet to be known. But what we know is that in our body, if we take DNA, 99% of the total DNA in our body is bacterial, and 1% is our own human cells. And perhaps it's a teamwork. What controls you? Is it your genes or is it your shit? Perhaps it is the teamwork, and I think it's that both are indispensable. What we see is just the tip of iceberg, and we still have to go a long way to uncover the microbial mysteries. And what we know now is that the good food plays a very good role in the in our healthy microbiome. So we must consume healthy food. These are the references. Somebody interested can read these papers, dedicated to late Professor Faisal Masood, my friend, my mentor, my teacher, and it was him that I got inspiration and to learn more about this gut microbiota. So you can learn more on Medurstand. This is my channel, Professor Aziz-ur-Rehman. Thank you very much for watching this video. Thank you very much.